Thank you. Right, so it's my utter pleasure today to have a representative from Aarhus University and here the Department of Food Science at Aarhus. I've worked with Aarhus for many years on different projects and I know the head of the department extremely well. If you ever get a chance to go to Aarhus University, go and have a look around. You will be envious of the setup, although Carl may tell us otherwise, but I doubt it. So today we have Carl-Otto Ottosen, and he's going to talk about the effects of light spectra plant responses. His area of research has been all around photosynthesis in plants and their changes in respect to abiotic factors, including CO2 and other things. I think the whole biochemistry and biology around photosynthesis is an extremely laudable goal because minute changes in photosynthesis, if we can make them to plants, can massively increase productivity, but we have to understand the biology. So we have an expert here to talk us through these systems. So Carl, it's all yours. Just give me a shout and I'll change the slides for you.
Yes, thanks for the introduction. Yeah, you're always welcome in Aarhus. We have just pre-COVID, we built new facilities and then we went home afterwards, but that's life. Next slide, please. So I was not completely sure who was the listeners today, so I will talk a little about what light is and kind of talking about the effect of light spectra in the greenhouse versus close plant production or vertical farms or urban farming or whatever you want to call it. And then some kind of perspective, how can we use spectra in greenhouses or protective systems, because that's kind of what the future of LEDs. Next slide. So when you start looking for LEDs, you get extremely frustrated. And if you press slide, next slide, it's really, shouldn't say that to my wife at least, but she's not in today, that it's like buying shoes. You never should never select more than 2 at a time, but often you end up with having tons of them with different specifications, different types, and it is a nightmare to sort out which kind of spectra you're using. It becomes even worse when you start looking at literature on LEDs. Next slide. And there we talked about light, and of course for plants we don't care about normally we don't care about most of the light, but we are talking about the light level in the power area, and in the end I will try to move a little to other areas too. But light is kind of a lot of different things. Next slide. It's just an example. It's actually made by Eva Rosenqvist from Copenhagen University, who did some measurements of spectral distribution of the natural light done in a garden. She measured the brownish one is kind of a normal daylight. The more darker brown is overcast. Then measured if you shade it massively or if you shade it by a leaf, cucumber leaf or a skimmia bush, which is a hardy ornamental here in the at least in Danish gardens or English gardens too. And just the change that you see during a day between plants under different plant leaves makes you a bit worried about how can we design a spectral distribution that can be used in a commercial setting in a greenhouse, more or less, which is where we started with using LEDs, probably as one of the first ones to work on LEDs at all in commercial settings. Next slide. So when we talk about light and photosynthesis, we started photosynthesis, and roughly the more light, the more photosynthesis. And the photons or the plants don't really care about the color of the photons as such, said in a rough way. But when we want to play, so in a greenhouse, we can put up LEDs and we can use the LEDs as a light source for growing plants. And since it's mixed with natural light most of the days, even today on a gray day, it's the color of the spectrum might not be so important. I'm coming back to that. But if you move into a closed system, then you need to worry a lot about how the different pigments are being affected. And if we take next slide. It's down there. So we mostly focus on, in relation to photosynthesis, we mostly focus on things in relation to sorry, I have two screens. I make a bit chaos here. So when we, if you take next slide to make it easier, if we look, could I have next slide? Thanks. If we look at kind of the spectral distribution also outside the PAR, you have all these different pigments or phytochromes which absorb light in different ranges. So if we want to manipulate a given process by a spectra, we can apply specific lights. But a lot of the experiments have been done on very specific, very narrow bands of light. And if you look at like the phototropins and cryptochromes, they are overlapping. So just a tiny change in the light spectrum might affect it. We had some experiments with green lamps, and some of them were bluish green and some of them were green, and we could see really large differences in responses in terms of pigments. Just this small change because you actually have a tail in the light spectrum. They are not very specific. So if we move on to the next slide, then we can see that we actually have most of the pigment reactions to photosynthesis are some kind of protective or absorbing mechanisms like the chlorophylls. But then we have the carotenoids, which also act as a, say, supposed health benefits as a secondary metabolite. They usually work in protection of the photoprotection of the photosynthesis. And then in the end, we have the flavonoids, the non-coloring ones, which mostly works as sunscreens, but also increase the effects of UV. So we have chlorophylls as a photosynthetic component, increasing the gas exchange, potential gas exchange, and then we have protecting components which also act as a potential health benefits for people. So we play with, when we play with LEDs on the color combination, we can actually make a lot of different variations depending on what we want to do. From an energy point of view, of course, we can't just add more light, even though we like to, because the cost of light has increased. Well, especially the last 3 or 4 months, nobody worries about having too much light. It's too expensive. But if we take next slide, then we'll see this is a picture from northern or southern Finland, actually, in middle of January. Outside is -24, inside it's around 20-ish. And in that climate, we can actually completely manipulate the light spectrum as we wish, because the day length in that period is 4 days or 4 hours or something like that. So in the controlled environment, in that case, it is a controlled environment which is affected by outside. A little only, especially in winter. It's the light internally in greenhouse is a dominant factor. So for greenhouse production, at least in the northern latitudes, then we can use the spectra in the dark period to manipulate. While during the middle of the summer or the late spring, early autumn, forget about it. The light, natural light will be important. We use that, next slide please, to look at some lettuce plants where we had an idea from early experiment that blue light might affect the stomatal regulation or increase stomatal regulation. But when we did it in 24 hours, we couldn't really see strong effects. More compact plants, but no effect on stomatal regulation. But then we tried to add blue light in different periods of day, kind of seeing what happens if you give it before dawn and after dawn and during the day or 24 hours. And this is 2 different lettuce types, obviously one reddish and one greenish. And there was not much difference in a dry way in that. The plants grow the same way. But then for the next slide, if we look at the stomatal regulations, next slide. When we look at the stomatal regulation, we see that the red Rosa had really no effect whatsoever, whatever blue light we did, but we had a strong effect of the blue light in Batavia compared to the control. The control is the white one. So the blue light had some effect. It tended to open the stomates if you gave them light early morning. So it's kind of a triggering effect on the plants. Of course, we would like to use that in commercial settings, but it might be more complicated. But if you'll end, next slide. Next slide, please. Oh, jumping too fast. Thanks. If we look at the metabolites, then the blue light increased metabolites very strongly in Lolloroso, but it had no effect whatsoever in Batavia. So we are actually, depending on which combination of blue and red we get, we can have a lot of different effects on the spectral, on the stomatal regulation and on the metabolites. And if you look at different varieties of lettuce, then you can get literally whatever value you want to have. And that's kind of a nightmare to work with. So next slide. We also did some experiments in late, this is done early winter, and this is an experiment we did in the middle of winter, where we applied UVA in a red and blue background, green in a red background, blue and white, blue and red, and blue and red in the normal standard light, LED light, these purple combinations, but with a background of natural light. And the background of natural light in Denmark in the middle of the winter is, say, 40, 50 micromoles. So it's not much. And then we try to look into, next slide. We could see that the difference is well, just an example of how the spectral distribution was made, where the grayish is the natural light we measured. And to make a long story short, next slide. We made a lot of different sizes of BASIL, but the fun part was even then small amount of UVA we were using managed to affect the flavanol index and the anthocyanin index compared to the other colors. So we can, even during the normal condition with natural light and normal artificial light in the greenhouse, affect the composition of the plants. One small detail which popped up in this, if you look at the next slide, was that the green treatment actually increased the shelf life of the plants. You all know that if you buy basil on Wednesday and they've been shipped from the production site on Monday, it goes crap on Friday because they wilt. And they've been shipped, normally they ship them with all other vegetables at 5 degrees, which is not really good quality, makes no good quality for them. But in this case, the green one actually had a better keeping quality than the rest. We have no idea why. We didn't really dig more into it. Next slide. When we then move towards LEDs and colors, this is a picture I stole from Bruce Buckby paper in 2013, just showing that you have different, white is not just white. You always know that when you buy the wrong LED lamp for your at home, sometimes they are white, sometimes they are very colorful. So here's an example. He's been growing radishes in different light combinations. You see the visual estimate that they are growing differently, but it also has some effects on the plants. And we did, next slide, a small experiment. Oh, jump that one. That was from the old series. We made a small experiment. We had some campanulas, small plants or plantlets, which we grew in a white lamp we were given as a demonstration from a company. So it was very bluish white. And then we compared it to rooting under red and white and red and blue, a Dutch company who produced those in large numbers. And you can actually see the plants in front of you are a bluish blue type campanula. The other ones are white, and the whites are notoriously poorer rooting than the rest. But it's very clear from the pictures that the red and blue, so the kind of 80% red, 20% blue, has the best results. So when we look at white broadband lamps, one has to be very careful and do some trials with different plant species first before you even consider buying. That is one of the challenges. Because normally, next slide, the most productions are made in these blue and what I call the purple lamps of 80% or 85% red and 15% blue. You can even have them in higher percentage of red because then they're more efficient. But the companies doesn't always tell. So it is one way of trying to make a system like for production. This is in a closed system where we really normally can pick your spectras. This side is though in old factory stories, so they have some natural light in. That's convenient because if you work in this blue and red environment, well, I've never asked health and safety what they like, whether they accept it or not, but that's will come sooner or later. We will need glasses. And there's a tons of, if you take next slide, there's a tons of experiments done in closed systems on trying to see how plants react. And this was a relatively recent paper I found in food chemistry for people who really hadn't been growing very much plants in a closed environment. They're using Hogerland, which is an old nutrient system, which is probably not used in many places anymore, but they show that, okay, if you add elevated CO2 and you add more nutrients, then you get larger plants. Good. We could have published that many years ago. And they also made, next slide, a small experiment where they mixed different combinations of light, but it's very complicated to see what is the effect because the pictures are not being color adjusted properly. So which one are the best of these combinations is, I find it very complicated to see. But typically we talk about white light, blue light, and red light, and white is then regarded as a full spectra, which is not always the case because you have to look at your spectra to be sure that is actually the full spectrum. Many of them are just very white with a coated blue, just like the picture of the campanulas initially. And we did in small experiments. If you take the next slide, this is obviously basil grown under standard conditions around 200 micromoles on the blue, red, and white. If you just press the next slide, you can see the names better. And the white one, well, visibly you see the red one is kind of bending the leaves downstairs. The blue is kind of little bendy, little paler, and the white has smaller, sharper, we say sharper leaves. And the next slide, we measured some metabolites, and you can see that blue plants have an increased amount of flavonoids because of the energy level from blue. It has less chlorophyll, even though it looks like okay. But the fun part of this, we did a tasting, and the blue one had a really prominent taste of turpentine or kerosene, whatever you want to call it. So even though they look the same, you can just by manipulating light composition, you can create different flavors. And it's, even though the white one has a high component of blue in it, it's still not linked the same. We did some analysis of it. So starting working with ADs and spectras becomes sometimes a bit tricky. If you take the next slide. This is just an example of the same setup with lettuce. And well, who would ever think of buying the red one while the blue one becomes a little reddish, the white one looks like a compact one. So there's really a lot of effects which we don't know too much about. And we continued the experiments, just mixing a lot of different compositions. Next slide. And then and you can see, you can literally design a shape, a color, depending on how you mix the colors and color combinations. And even the far red, which is left to the left, my left at least, becomes long and thin, as you would expect with the far red, while the red ones, the far most right, is soft compared to the other ones. So mixing lights becomes funny. And for the next slide, we decided that we wanted to do more on UV, and then we moved into UVA in this case, also like similar to the first experiment with BASIL. And then there we used LED sunlight, which is a control, which is this broadband LED lamp. So we have red, blue, and white plus UV also for them. And the next slide shows that well, there's a major difference. Green and red has a really negative effect on the size, and the sunlight, the broadband white, is the best one. So again, we can get a lot of different variations just by adjusting our light levels. Next slide showing the leaf area doesn't show the same amount of variation, but still the green and red had a very negative effect on the leaf size or leaf area. And just a picture, next slide is a picture of the plants, and you can really get, they're a bit too big, but the green and red is reflected, the data we saw before is really not very well functioning. It was teasing us a little because we've seen some earlier data where the green was actually quite good in relation to the performance. So we made a PhD project, next slide, where we wanted to focus on UV. And we know UV has a lot of different effects on plant growth, both, well, in small degrees, it can promote the chlorophyll of flavonoids, as we saw before. It will often be some kind of reaction, protecting the plants as much as possible. But in small amounts, we can actually trigger some components in the plants of the shaping of the plants. So next slide. We decided to kind of look at a mixture of UVB in a background of these monochromatic colors. Very artificial, but be able to see is UV having some interaction with when you use these different color combinations. And the setup experiment was shown in next slide, where we simply grew the plants in the red, blue, and green and white, broadband white, and then we added UV for a period after some days, and then they had 6 hours of UV during the light period, and then we gave the plants a lot of high light level, like 1,600 micromole, just to see, do we have any changes in the FEFM? So is the photochemistry being affected by having these combinations of light? Next slide. Well, now I have a different range of my slides versus your slides. That's a problem. Plant biomass, blue and green blue made the plants more compact, but the green, in this case, in basil, not in lettuce, had more biomass and a faster leaf development. So it kind of is reflecting the contrast between species that, in this case, we saw a very strong positive effect of UV, of the green. UV normally decreased biomass. And then we take next slide. Just an example of this, how the plants look like. The red is what we expected, just like we saw in the lettuce. Your plants try to avoid it, get overexcited because the red doesn't penetrate through the leaves. The blue has more light capture. They are being protected, of course, a little by increased flavonoids. The white one is very compact. It is quite a lot of high light level for the plants, and the green appears again, just like the data showed, to be quite strong effect, positive effect. They actually look like the best plants. And if you take next slide, we looked at the gas exchange for synthesis, and well, there's not much difference between blue and white. The red is non-functional, less functional, and while the UV combined with the red increased the photosynthesis. So strangely, we see some interaction with UVB and red, but nothing with green, and some strong with blue and white. And again, the white ones has a strong component of blue. So it is sometimes a bit complicated to explain what's going on, that we have some of these very strong effects coming up. And if you take this last slide in this round, we are looking at the FEFM, so the quantum yield, apparent quantum yield of plants, and there's not much stress in the first series of experiments, but we see it when we get this high light levels after the treatment. Some of these lines, like the blue and greens, becomes more sensitive. There's a change there's a decline in the FEFM. So we also did some very rough metabolites, and not surprising, similar to what we've seen before, white light has the highest content of chlorophyll and flavonoids, but UV increased chlorophyll in some cases, and it increased flavonoids in other cases. So we see a lot of complex reactions, and we've been looking at the metabolites more in deep, kind of targeted metabolomics, which I don't want to go into here, but if you look at the papers, we have 2 or 3 papers around this, authors by Carolina. So from these experimental UV on cucumbers, we saw that this monochromatic light background has a very strong effect on plant growth, actually more than UV. So the UV secondary effect that we had hoped to see was not so pronounced. We don't see the same amount of pigments, so there's some kind of negative interaction or interaction between colors and the UV. But like the red and UV has some positive interaction, and we're discussing whether there's some kind of priming effect when UV is in combination with other stresses. So that's kind of some of the experiments we've been running recently on UV and interactions. But we've also been playing at other combinations. If you take the next slide, we jump this one. This is just an example that we tried. Bruce Buckby and others have been playing with thinking about far red. Could it have some effects on photosynthesis? And we did an experiment with cucumbers and where they're grown in the white light, and then we have far red, 10% far red in a white background. And the difference between the two pictures is 4 days. So we put it in on Friday. On Monday, we had to take them out. Otherwise, they would outgrow the chamber. So for cucumbers, never, ever think about having too much far red, because then you would just have a strong elongation. And it doesn't fully correspond to this slide from, next slide, from Bruce Buckby and his colleagues, where they claim that if you add far red in combination of other light sources like white lights, it has the same effect as adding more light levels. So if you look at these different species, the blue light is the white light, and then the triangles, the red triangles or circles are white plus far red, and it adds on top on the light response quite nicely, and depending on leaf area index, except in spinach where you don't see any differences. But please note, it is actually at light levels that is beyond 500 micromoles, which is relatively high light levels that you're working on, and which you probably never would try to grow plants under, at least unless you have too much money. Mark Earsel's next slide. Oh, next slide. I hope. No, reverse. That's the disadvantage of changing slides and not having the right version. It's just some examples that we also tried this far red effect on tomatoes. This is a very compact tomato type, and we could see that there was really large difference in playing with blue-red combinations and also with far red. And the far red in these tomatoes actually promoted the growth, and it strongly promoted the development of flowers very quickly and fruits. So you actually can see by manipulating the far red in some species, it works perfectly, but in a cucumber, well, it just makes it a very long plant. So next slide, I hope, yes. So LEDs is really a challenge. When you buy an LED lamp from the kind of large number of Chinese suppliers or European suppliers, there's no standards, because people tend to pick LEDs or which are suitable for their lamps in technology, and we can't always compare between two different types, because there might be a minute or larger difference in the wavelengths they're working on. So often we're also missing any information on power output, light distribution, spectral distribution, how uniform it is. So it's not so easy if you want to look into and buy LEDs for experimental purposes. And I think in many places of the world, there's tendency that any engineering department can make their own LEDs, which physiology is then being using later on. And it means that it's very complicated to compare between experiments done in different locations, because they might be homemade. And one of the biggest challenges we have with LEDs right now is to get them repaired. If they break down, they are not really complicated to repair. You have to more or less replace the power box and the whole print box. So the question in the end is, how much can we use the spectra in the real life? If you want to have a production, or we can run experiments, but if you want to upscale it and run a practical experiment producing plants. Right now I'm writing a report about vertical farming for the Danish Ministry of Agriculture, and it's not so easy to come up with good solutions. But next slide. So the biggest challenge is to translate our many papers on LEDs to the real world. And one experience I had presenting some data in Max Planck Institute in Germany, one of the Max Plancks, many scientists are really reluctant to change the light sources in their climate chambers, because we've been using mercury lamps for 25 years. Now we move to LEDs, but it will change. So how can we compare our gene expression from now to earlier? And of course, all types of LEDs has less heated output, so your leaf temperature will change. And one thing that they had noticed that was some of the stress expressed genes were less pronounced now when they moved to LEDs. But it's in many cases, we could move to broadband LEDs if you check that they are really broadbands. And we've now on our third generation of LEDs in controlled environments for that purpose. But one thing which isn't convenient, if you want to work more with LEDs, we can make much more dynamic conditions. So you can make a simulation of a day in terms of at least light intensity. If you have too much money, you can also add parameters, so you can have light distribution changing or spectral distribution changing over a day. And that means, in our case, for greenhouses, that if the light level is high outside, we can actually dim the lamps, or if the light level is low, you can keep a more stable condition. So you can use more dynamic conditions, which has become very a challenge to work on in terms of physiology, how plants react. Because in nature, light is always dynamic. So the next slide, almost the last slide, sorry about the time. For multi-layer systems or vertical farming, we see that there is a possibility to create a lot of additional values by selecting LEDs. So you can have more compact plants like the tomatoes. You can also have less compact, like the cucumbers, and you can have differential taste. But moving between species, moving within varieties, it is a challenge. You can't always compare. And if I look now, sitting and reviewing, people have been running experiments with lettuce from anything from 50 micromole to 500 micromoles of light. And you see in some cases very strong growth effects, but you have less nutrient effects because the plants might outgrow the nutrient amounts if you don't adjust it. One thing we I think will be important if we want to develop vertical farming, is that we have to play with the spectral composition in different phases. So apply different spectrals initially during the main production phase, and then potentially add another spectra towards the end to increase the secondary metabolites and the health benefits. One of the biggest challenges right now, we are at 3 moles per micromoles per watt. And of course, it would be nice to have more energy efficient lamps, but there's also the challenge of the lamps are really become still very expensive compared to what I would expect it. I'm really missing some disruption in the prices of LEDs. But it's again a dominant market by very few companies. So that was just some kind of snapshots or ideas of what we can do spectrals for. If you take the next slide. So this is kind of a experiments we've been doing in Denmark in different projects, but we also run two indirect projects, one with including Lincoln in UK, and then we are linked to our national projects, Innovation Food, so now it's called SciFood, which is working on food research. So this is my words, and we can take the next slide. So it is confusing to work on, but still I think some curiosity is needed. Any questions? And thank you for listening.
Thanks for that, Carl. I think you've generated a few questions for me anyway, but I'll let others have a go first. Anyone got any questions? Rob.
Hi, Carl. Thanks for that presentation. Very interesting. So, I mean, I think I'm right. So, I mean, for me, what's really interesting, right, is the effect of spectrum on photosynthesis, right? Am I right in interpreting your slides correctly, right? So your plants were grown under different lighting regimes, and you then did a simulation light intensity curves, right? Mm-hmm. And depending on the regime, you saw a higher maximal rate of CO2 assimilation.
Yep.
So, but I didn't see anything about stomatal conductance. So was that high CO2 assimilation associated with a higher stomatal conductance, or is it other factors that's leading to that?
In some cases, like the green light, we don't see any increase in stomatal conductance, but we can only assume that you have a better penetration of light, the green light, deeper into the leaf. So you have a larger absorption of the green light compared to the blue light and red light. In red light, you tend to see some stomatal regulation, but not limiting the gas exchange. But again, what you see with the red light is that it penetrates very shallow in the leaf, and then you see some kind of light avoidance, this obviously cup-shaped reactions. For blue light, clearly the link to stomatal regulation. Okay. So it depends on the color.
All right, thanks.
Raul.
Yeah, very nice presentation. Thanks. So you show that control environment condition, require the correct selection of light, but how much progress have been made in the selection of the cultivar or varieties for different crops? Because so far, maybe most of the varieties have been selected off-site, right?
The simple answer is nothing.
Okay.
There's a paper from Wageningen where they've been looking at rills from tomatoes, and they could see they compared mercury or sodium lamps to LEDs, and there was some difference in the growth rates, which I think was associated to leaf temperature rather than any genetic components. But nobody has been breeding varieties for closed environments, to my knowledge. Well, I have 30 centimeters of paper behind me. Fortunately, you can't see that.
Thank you.
I think there's actually some companies getting into that now. I mean, you've got Unfold in California trying to get into that, but I think they're basically trying to monetize the old seeds from Bayer that couldn't grow outside very well. whereas they see indoor for vertical farming probably an easier market. So these are the ones that are probably incredibly disease susceptible. In a controlled environment, you kind of don't need to worry about that. Well, I don't think that's really breeding, but they're starting basically to broaden the germplasm to grow in there.
Well, that's true, because in a vertical farm or in a closed environment, you don't, in principle, have to worry about biotic or abiotic stresses, because it should be clean. On the other hand, a lot of our experiments show that if you run some kind of dynamic in your climate, fluctuation temperature, fluctuating light, then you get more secondary metabolites.
Yeah.
And that would be favorable. So I don't know what the breeding target would be for plants or genotypes for vertical farming. Compactness, of course.
Yeah. I think it's changing the architecture. Maybe if you're feeding them so well, diminishing roots. So less goes into the root biomass, because unless that's what you're growing for.
But most plants seem to have some kind of fixed ratio between top and shoot. But it might, one of our first experiments with roots was we grew cucumbers in limited soil. Yeah. In the end, we grew a 2-meter cucumber in 100 milliliters of container, because we added enough nutrients and water. So perhaps you can limit the roots by having enough nutrients and oxygen for the plant. It looks weird, yes.
I can imagine. Jean, you have a question.
Hello, that was a really interesting talk. I know most of what you were talking about was obviously related to controlled conditions, but I just wondered, when I was thinking about not growing right outside, but like in polytunnels, whether different plastics lift different light. I mean, obviously not all the light's coming through the plastic, but I just wondered if there was differences between different plastics and what light is let into the plants.
Most of the changes, there has been a lot of spectral attempts to use filters in polytunnels, especially in France, to manipulate. But the challenge is that if you put a filter, you also reduce the light level. So we haven't been working on that. We want to have as much as possible. But in most cases, the UV is the factor that you can play with, or you have to look into. I think Lancaster did some experiments where they had slightly UV penetrating plastics, like 7%. Oh, can't remember. And that showed that things like lettuce were more compact when you had them in the block. They could actually be taken from the polytunnel and moved directly into the field. They didn't have to have any recuperation period before being replanted. And things like thyme had a lot more oil in the leaves per plant. So even though this was in a protected in polytunnel with UV penetrating, it had a strong effect on the plants.
Thank you.
Welcome.
One thing strikes me. I mean, it still looks kind of like the Wild West with regard to this plant does this with this light recipe and this, well, even variety of variation you get. It rather sounds like, from an application basis, it's crying out for collect all the data and kind of use AI to try and predict and allow that to use AI to start programming in things like dynamic lighting and things like that.
Yeah, but if you can get one thing you could do is do a meta-analysis using AI, but also basic meta-analysis. But the challenge is, when I look at some of the literatures, there's not enough information to really, let's say, repeat the experiment.
Yeah.
Because if you have, like, some of these lamps are 400 nanometers in blue, and if then they are 420, how much can we compare the 400 and 420? Because they still have this a tail on it. As I mentioned, the green lamp we had, the bluish green and green lamp, really had a difference in metabolites.
So it almost seems like, certainly for when you're reporting on this in a paper, we need almost like a Miami convention so there's say anything with the LED lights has to be much more specific in its reporting. So if you've got LEDs, you have to almost dig out what the manufacturing guide is, or you measure it yourself to really define what that LED light is giving out.
Yep, that's the way we start when we get a lamp. Put in and measure.
Cool. Any more questions from anyone else? So thinking back to, you were talking about earlier, you're reporting for the Danish government. We're kind of doing something similar for the Scottish government. Now, in countries that have got significant renewable energies, can you see them become winners in terms of LED-driven controlled environment systems?
Does any country have...
particularly on farm energy? So if the energy, you've paid for the capital, you have the energy, the system effectively can be classed as free energy now.
You know, if we regard green energy or non-fossil energy as free energy, then of course the CO2 footprint from vertical farms go down. But in Denmark, right now the government's way of looking at energy is the amount of energy used per square meter, and disregarding whether it's green or blue or what color it is. So right now the CO2 footprint from, well, Orsini in Bologna has made some very nice graphs showing that vertical farming equals the CO2 footprint like beef. Perhaps a bit rude way of saying it, but there's a very fresh analysis by a guy called Bloom and his colleagues has done looking into vertical farms, field and open and closed greenhouses in the Netherlands. And you say 2 to 3 times higher CO2 footprint compared to in a vertical farm. Mm-hmm. So it's not a really pitch to sell it from an energy point of view.
Yeah.
And you could also say that the greenhouses, my colleagues in Serheim in Norway has just designed a greenhouse which has completely, it's a completely gas-free greenhouse with a CO2 capture. So it's CO2 neutral. And some of the growers I work with in Denmark, they are moving into heat exchangers, which means they can move from gas or whatever they're using. So the development might be beneficial for both partners. The biggest challenge I see in vertical farming is there's no standards.
Yeah.
Everybody's, it might be a rude way of saying it, but everybody is developing their own setup standard, which makes it very complicated to compare. At least in greenhouse, we use glass, we have some amount of aluminiums, and you have a control system, which is, well, you have 4 companies in Europe who are developing climate control system, major companies. In vertical farming, it is, to me, a mess.
And I would agree. I think there's people working within that. I think with ones we know, they kind of agree as well. But I think we're all waiting on someone to blink and start it. So, I mean, I think we certainly are going to try and initiate standardization. Whether there's some certification, I mean, certainly one of the other arguments, maybe it's not so bad in Denmark, is here a lot of the stuff from the vertical farm or even hydroponic glasshouse could arguably be classed as organic, but the organic lobby clearly wouldn't allow that here. So there's an element of certification or a different classification may come in should they want it. I'm not convinced it's needed, to be honest. People will either buy it because they like it or they won't.
Well, the challenge that some of the pitches being used for vertical farming is space. We don't see the space as a major problem in Denmark. Talk about Shanghai and so on, it might be okay. In terms of water and nutrient and pesticides, they use roughly the same as a modern greenhouse doing herbs and spices. And then the idea of local, there's 2 companies in Denmark, larger scale vertical farms, and they ship to the supermarket. So they go to the supermarket chains, which means it's not all of Denmark is local in that case, because you can go to the supermarket everywhere in Denmark.
Yeah, it then becomes some are more local than others. Yeah.
So I find it challenging, but the reason for writing the report was they would like to have subsidies, and I struggle to find arguments for giving them special subsidies.
I think only, maybe to finish on this, certainly here, one of the arguments is, and this could apply to probably many of the glasshouse systems, is in the UK anyway, you get a subsidy if you're buying diesel or tractors. They're arguing, why not have a subsidy for the energy in glasshouses or vertical farms, because they're using electricity. So can you produce subsidized electricity for food production? I don't know if you've got an analogous system in Denmark, but here that argument is kind of increasing, because it's almost like one sector is more subsidized than the other. Although I think living on subsidies is never a great idea.
Well, what the strategy in Denmark is that you can get subsidies for investments in resource-saving methods, so less water, less nutrients, less pesticides, and less energy.
Sure.
So if you want to replace your screens in the greenhouse, then you get some subsidies for that.
Cool. Okay, well, thanks for a very informative talk. I'm sure we will be meeting again soon as we start to develop our phenotyping centre and discuss more with you on yours. But if people can show their normal appreciation, and thanks for that.
You're welcome.
It was good. The talk will be online, shouldn't be too long, and we look forward to speaking to you again. Hopefully we'll see you all at the next APGC seminar, folks. Bye.
Bye.